So just to recap where we left off on Tuesday, we were differentiating the structure, not really the full structure, but we were talking about some important structural features of NADH, FADH, and then also ATP. And we kind of concluded that NADH and FADH are going to be used, or alternatively being oxidized and reduced to basically pick up or release hydrogen ions and electrons. So we are going to see them produce stepwise, some of them, procedurally in some of these metabolic processes, but we won't see them put to work until we get into something called the electron transport chain. Okay, so right now we're just kind of introducing, if you will, the players in the game. We also introduced a couple of terms, including phosphorylation, oxidation reduction as well. What about phosphorylation? What does that mean? Adding a phosphate. If something's phosphorylated, it just gained phosphate. That's technically what's happening. And we said functionally, really, something if it gets phosphorylated, it gets what? Not add energy. It gets a form of energy. Okay, we're giving it something. Right, we're anabolizing it. We're transferring into energy into it. So what I'd like you to just kind of loosely connect is if something's phosphorylated, it's getting energy. So technically, it's getting a phosphate group, but that's transferring potential energy in there. And if something gets potential energy, it could do some form of work. Alternatively, if something has become dephosphorylated,
You lose energy and lose a phosphate.
Lose a phosphate, transfer out the potential energy of that molecule. Very good. Is that concept okay? Not too bad? So we will see ATP alternatively phosphorylated and dephosphorylated through the semester. So we're going to make it more energetic or a little bit less energetic. ATP is this the only thing that could be ADP, is it the only thing that could be phosphorylated? No. Okay. Pick your favorite protein. It can probably be phosphorylated. You may have a favorite protein. Not yet. One day you'll have a favorite protein. All right. And we went ahead and said, well, that phosphorylation can really occur by one of two primary methods. One method is referred to as substrate level phosphorylation. And what characteristics do we give for substrate level phosphorylation?
Very fast and precise.
It's very fast, it's very precise, and it's direct. That phosphate is directly transferred from one substrate to another. So it's coming from one substrate to another. The analogy that we gave is this visual here with the use of glucose. And glucose is shown in the green structure. And during this process that we will become familiar with on glycolysis, the first thing that happens with glucose is it's phosphorylated by ATP. So we're not showing the structure of ATP, we're just little flash is representing ATP. So ATP can phosphorylate glucose. We'll come back to that. If ATP phosphorylates something, it has just become ATP itself has become it turns into ADP because it has just become dephosphorylated. Okay, so if I take a phosphate off of it, I have dephosphorylated it. But if you notice in this image, the phosphate is now going to be bound to our substrate glucose. So the phosphate represented here was not originally bound to glucose. We phosphorylated on the 6 carbons. Now we have glucose 6-phosphate. So it's directly just transferred, goes from here to here, from one substrate to another. Do we list which of the two metabolic pathways that we'll study where the substrate level phosphorylation takes place? phosphorylation during glycolysis and also during the Krebs cycle or the citric acid cycle. Citric acid cycle is the same as is another name for the Krebs cycle, and another name has a third name. It's called the tricarboxylic acid cycle. And, if you're a biochemist or however you want to study it, but they're all the same pathway, different names, same thing. Good question. So substrate level phosphorylation, there's a direct transfer of a phosphate from one substrate to another. It's very direct, it's very precise, and it's quick. It's usually just controlled by an enzyme, like for example here, an enzyme called hexokinase makes this glucose phosphorylation. So it's very precise and specific. By contrast, another form of phosphorylation is referred to as oxidative phosphorylation. This one, you kind of take the talking points that I mentioned about substrate level, then you can say, well, it's probably not as direct, maybe not as fast or quick, and because it's not as direct, it actually might be more wet. I don't know about the stability of it.
Can mess up.
Well, I think we could say that with everything though. Everything can mess up. What? Y'all are going down the negative pathway. I want you to go down the positive pathway. The glass is full. Do it. More efficient. It's going to be more efficient. What that means is, instead of transferring just one phosphate, I might be able to phosphorylate dozens. Okay, so dozens of phosphorylations may happen following around the oxidative phosphorylation. Furthermore, because it's not direct, the phosphate isn't coming directly from another substrate, and so the phosphate comes from in what's considered to be the inorganic phosphate. So you would write that as a large P and a subscript I. Inorganic phosphate is going to be your source of phosphate or oxidative phosphorylation. What that means is it's not bound to something like ATP. It's not bound to something like glucose, fill in the blank. It's just a phosphate group.
So that's going to be the substrate.
Oxidative phosphorylation uses inorganic phosphate.
It just uses.
It uses inorganic phosphate to phosphorylate upwards of dozens of molecules. We're going to see this in only one of our pathways. Which pathway will we see this today? We're going to talk about three. We listed two, so the last one is? The electron transport chain. Very good. So at the very end of the electron transport chain, ADP is going to be phosphorylated by oxidative phosphorylation. And depending on the quality of your vision, you may or may not be able to see that little inorganic phosphate. Like I tried to illustrate for you to write. Large P, subscript I, meaning it's just the phosphate group. It's not bound to carbon, hydrogen containing something. And so we're going to find a mechanism to push or force that inorganic phosphate onto ADP to overcome that electrostatic repulsion to make what? ATP, adenosine triphosphate, and a bunch of it. Because we said it's efficient. It takes a little while to get there, but payoff is good for the cell. Questions at this point about the difference between substrate level versus oxidative phosphorylation? Alrighty. We're going to follow a very narrow path for ATP synthesis, and we're going to look at the oxidation of glucose. Now, what does that statement mean? Glucose is losing or giving up its electrons. Okay, so we are going to metabolize, oxidize, break down glucose for its what? Energy. We're going to break its bonds to strip off electrons. And so we're going to see electrons being removed. We're going to see hydrogen ions being removed. Carbon dioxide liberated. There are lots of inputs and outputs are going to be going on. If you recall, we mentioned that we want to oxidize nutrients like glucose in a more efficient manner. And so we want to do this very procedurally. We don't want to break all of the bonds in glucose simultaneous. We made an illusion that that would release too much energy into the environment, heat energy, and it would be lethal to a cell. So we don't want to kill our cells to eat, to power this, right? It would be futile. And so we need to procedurally metabolize or oxidize glucose. And that's what we're going to go down this narrow path. But do we need only a diet of sugar? Okay, want or need? Right, do we need it?
No.
No. So can we only oxidize glucose for nutrient, for ATP synthesis? No. I mean, we could go through in your nutrition class, your biochem class, you could go through and say, well, let's look at oxidation of different proteins or amino acids or lipids or whatever, right? You could go that way, but we're just going to say, what's the common, most familiar pathway that we can kind of consistently see in biology? So we're going to look at the catabolism, oxidation of glucose. If we look at glucose, does anybody know how many carbon atoms you would find in this? 6. 1, 2, 3, 4, 5, 6. It's got 6 carbon atoms. And we're going to, first step is just chop it in half. So it has 6, we're going to chop it in half and put it into what? 3 carbon containing molecules, how many? 2 of them. So we're going to chop it into 2, and each thing is going to have 3 carbons about it. So if we kind of put it in hopefully visual perspective of what's going on. Our nutrients, right? We're not photosynthetic, so we can't make our sugars, right? We have to go out and do what? We're animals, so we have to go out and eat, right? And when you eat, we don't eat exactly what we need. We don't eat continuous. Most people don't eat continuously, right? We go through a period of fasting when you sleep or whatever. So at any point, you're probably going to have excessive nutrients in your digestive system, right? But does your body just, so let's say you have a Twinkie diet problem, right? You have to eat Twinkies all the time. And if you read the calorie count, you know they have lots of potential energy in them. And so if you eat, say, 5 Twinkies for each meal, so you get up to 15 Twinkies a day, what's going to happen to your nutrient balance? What are you going to do with all those things? Store it. You're going to store it. Okay, you're not going to probably...
You're going to store a lot of fat.
You're going to store a lot of it, right? It's not going to just enter and then you enjoy it and then it goes out, right? Your body says, thank you, I need this, thank you, so it banks it. The nutrients in your cells as fat or glycogen in your liver, okay? So this is what this image is trying to kind of conjure up. You take in nutrients, when you take them in, they don't stay in your digestive system, they don't stay in your stomach indefinitely. The point is the digestive system there is just kind of start that catabolic process and then mobilize it into your circulatory system. Your circulatory system also isn't a source of storage. So once the nutrients go into your blood, they're there just to be distributed to your cells to either store, like we just alluded to, or what? Either store it or do what? Use it. And so depending on which tissue you're at, for example, in your liver, you can store carbohydrates. This one called glycogen, or your adipocytes could store lipids as fats. Your skeletal muscles could store protein as proteins, amino acids as proteins. So we can store nutrients. But at some point, you're going to have to mobilize those nutrients for the rest of your cells in your body, like your brain, your nerve cells in your brain, your spinal cord, everywhere in your body. Your cells need to use those nutrients at some point. And that's where we're going to pick up. After the nutrients have been mobilized, what happens to them? And we're going to hone in on glucose, not the rest of our potential nutrient sources. And so the first thing is glucose, once it gets into a cell, so this yellow beige area is representing a cell. Okay, so pick your cell of choice. Maybe you want to draw a membrane around it to represent that you are in a cell. So glucose is transported into a cell's cytoplasm. And if that cell is not going to store it, it is going to catabolize it. What is the name of the catabolic process of glucose oxidation? What do we call that? Glycolysis. Glycolysis. Let's work through the word backwards. Lysis tells us we are splitting, breaking a sugar. So we're splitting a sweet, right, a sugar, such as glucose. But let's review. Where is this happening? What part of the cell? We've gotten pretty technical. Cytosol. In the cytosol of some cell. This is really important where the location is. Okay. Glycolysis under enzymes. Each step is enzymatically regulated. We're not going to go through each of the steps. I'm trying to condense this as best we can into a day and a half discussion. So glycolysis happens where glucose is split. It's regulated by enzymes. And then the end product is what?
2 ATPs and a pyruvic acid.
Okay, so we got some ATP. Let's go ahead and write that down since it's not on there. Right? So following glycolysis, in addition, put a number here, 2 pyruvic acid, or pyruvate. Slight structural difference. 2 of those are made, and 2 ATP are made, net ATP are made by what type of phosphorylation? Substrate level phosphorylation. What else is released following glycolysis? 2 NADH, and NAD can't pick up 2 full hydrogen ions, it picks up part of them. So what else is liberated? A couple of hydrogen ions, or you could say protons. So 2 pyruvic acid, 2 NADH, 2 hydrogen ions, 2 ATP. Anything else? Water. Is water given off in this process? I don't think so. I can't recall it. Carbon dioxide is not given off yet. I can help you here. How many carbon does 6? Never work on Jeopardy. Glucose has how many carbon atoms? 6. And we said it gets broken into 2 3 carbon things. So 3 plus 3 is 6. So we've not lost any carbon yet. When it comes off, when the carbon atoms are removed, it comes off as CO2. So we haven't decarboxylated yet, but we will. Very good. So glycolysis happens where?
Cytosol.
In the cytosol, cytoplasm, where we essentially split glucose into 2 pyruvic acid molecules. ATP is made, NADH is produced, and a couple of hydrogen ions are also produced. But we just split it in half, so where do we have more potential energy that we could work on and break apart? Pyruvate. Do what? And the NADH. Okay. And so the NADH and the hydrogen ions will be used later. We'll talk about it in about 10-15 minutes. So we'll use it. It still has some. But more immediately, we can process what? The pyruvic acid. Notice there are 2 fates for pyruvic acid. 1, 2. There's an either or, one or the other. Okay? And why don't you put maybe a squiggly line or something to kind of say, well, maybe this one will happen, the offshooting to the right. Maybe that pathway will happen if what? if no oxygen or little to no oxygen, if our oxygen levels are below what?
Normal.
Normal set point. If oxygen is below set point, then pyruvic acid is going to go this way, which maybe you've called fermentation. That's a big if. So if oxygen is not available, pyruvic acid could be what? Fermented to produce lactic acid. So I don't want to talk about that so much yet, as long as you're trying to understand the fate depends on the status of the cell. Is that okay? So I'm going to presume your cells are happy, you've got good oxygen levels in your body. Okay? If oxygen levels are adequate at or above set point, the pyruvic acid can be transported where? Physically into the mitochondria. Okay, so physically it would go into the mitochondria. And maybe it would be helpful to represent the double membrane here. You can kind of see a slight coloration, but maybe want to draw a double membrane there. You know, that's going across the outer membrane, through the intermembrane space, across the inner membrane, and into the actual, what? The matrix of the mitochondria. If oxygen is readily available, pyruvate will not ferment, but it is physically transported into the mitochondria, getting ready for the Krebs cycle. But something really important has to happen first. Not shown here, not shown in this image. What happens first?
So it yanks up.
Pyruvic acid gets what?
Converted into acetyl CoA.
Acetyl-CoA. That pyruvic acid gets converted to acetyl-CoA. I don't know if I can write on here or not. I apologize in advance. This is hard. Wow. All right, that's close enough. Oh, well, sorry. You gotta hopefully you can get the gist. Once pyruvic acid is transported to mitochondria into the matrix, it doesn't immediately initiate the Krebs cycle. We have to convert it. That's where we're gonna see a decarboxylation and pick off some carbon. And then once it's formed to acetyl-CoA, then we can drive the what?
Krebs cycle.
The Krebs cycle. Before we look at the specificities though, how many pyruvic acid get transported into the matrix based on the original glucose? 2. So by all technicalities, the Krebs cycle happens twice per original glucose molecule. So each pyruvic acid gets converted into its own acetyl-CoA, which then could power its own cycle of the Krebs cycle. You will almost always see the Krebs cycle like this, like a roundabout or a Ferris wheel or something. But I don't want you to think that the molecules are in a whirlpool in your mitochondria, because they're not. It's just a cyclical pathway. What initiates a pathway is what ends the pathway. Okay, so we're going to process this molecule. Okay, so it's kind of continuous, cyclical, as the name implies. What are some important byproducts of the Krebs cycle? We have some NADH. So y'all want to look at it per term, or do you want to look at it totally? Let's look at it totally. So how much NADH would collectively be produced following the Krebs? 6 NADH and their respective hydrogen ions. 4 CO2. y'all are on it. Alright, so I'm gonna back up, repeat. We have how much NADH? 6. And their hydrogen release protons. How much carbon dioxide? 4. 2 FADH2. 2 ATP by which type of phosphorylation? The substrate level phosphorylation. Did we get everything?
6 hydrogen ions.
So the 6 hydrogens lumped in with the what I said, the NADH and their hydrogens will be 6. Very good. What else was added? I think we got everything, didn't we? Is there any other molecule besides the NADH, the FADH2 that we can continue to work on? At the end, like we saw at the end of glycolysis? Not really. We've picked off our hydrogen, NADH, and FADH2. Okay, so what that means is now we can use NADH, FADH2, and the hydrogen. So following the Krebs cycle, all of those FADH2 and NADH will be used in the next series of reactions referred to as the what? The electron transport chain. The electron transport chain occurs where? Along that inner mitochondrial membrane. We're going to see a transfer across that membrane from the matrix to the intermembrane space to the matrix. So we're just going to be fluxing back and forth between the two fluids of the mitochondria. There's going to be a lot going on during the electron transport chain. As the name implies, we're transporting electrons. We're making what?
Energy.
We're making
ATP.
We're making ATP to clarify. So we're going to synthesize some ATP. What else is made? We're going to create a gradient so we can create ATP by what type of phosphorylation? Oxidative phosphorylation. What are some byproducts? In addition to ATP, what is liberated?
NADH.
Well, we use the NADH, so we would release NAD. We would also release some FAD,
some water.
water,
Energy.
ATP. Oxygen is not made. We don't make oxygen. It's what?
It's water.
It's used. We use the oxygen. We don't make it. Which makes sense if you think about it. We need to breathe oxygen because we can't. We're not. I still wish we had a photosynthetic patch. I'm gonna engineer us. So you can go for your lunch break and eat some lawn and do what you do. That would be the most ideal. And cheaper, right? Free lunch. All right, so sorry. That brings us to something though. Some of these pathways may be referred to as aerobic or anaerobic. What does that mean?
Uses oxygen or doesn't use oxygen.
Either uses or requires or doesn't require oxygen to accept electrons. If something is aerobic, it requires oxygen to accept electrons. Yes. Because oxygen is the terminal or the final electron acceptor, that makes which process aerobic? Oxidative phosphorylation. The electron transport chain is most specifically which type of respiration? Aerobic. The electron transport chain is aerobic. What about glycolysis? If you've got the pathway open in your textbook on page On page 75, you look at the glycolytic pathway. You ever see just oxygen? Or you can look at it super simplified on page 76, figure 3.15a. Do we see oxygen as an input? Oh no. That's good. I was talking about photosynthesis patches anyway, so it's. Dang it. I have more trouble with this thing. Okay, thank you. All right, so what about glycolysis? How would we categorize it? Aerobic?
Anaerobic.
It is anaerobic. There are no oxygen molecules serving as the electron acceptors in this pathway. We don't see it as an input, right, picking up electrons. Here's where I'm going to caution you, where it's easy to get confused as a student coming from one class to another, is just because it doesn't require, a pathway doesn't require something, does that imply that it's not present?
No.
No. Glycolysis does not require oxygen to proceed. It does not indicate the oxygen levels in your body. So if you have adequate oxygen levels, you are still going to carry out which process? Glycolysis. It just doesn't use it. Okay? Does that make sense? What happens after glycolysis really is dependent upon oxygen, though. The fate of the rest of the reaction. If oxygen's available, then what? We can proceed. So pyruvic acid has to get transported where? Into the mitochondrial matrix. Then we can carry out the intermediate step or the linking step, and then we can carry out the Krebs cycle. The intermediate step. How can we categorize it? Aerobic or anaerobic? It would either it does or it doesn't. This one isn't quite ambiguous.
Anaerobic.
It is anaerobic. We do not see oxygen serving as an electron acceptor. Doesn't need it. The next phase. Krebs cycle. Aerobic or anaerobic?
Anaerobic. It doesn't require oxygen.
It is also anaerobic. It is also anaerobic. It does not require what? It doesn't accept oxygen. It doesn't use oxygen as a what?
Electron receptor.
An electron acceptor. So if you look at your Krebs cycle, if you look at all the inputs, you will never just see atmospheric oxygen entering the pathway. Okay, so the Krebs cycle is anaerobic. The only aerobic pathway that we're going to study is the electron transport chain. So you should be going, what? Am I doing that right now? What? Why? You just said if oxygen is not available, then blah, 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 right?
Oxygen's always there, it's just not used.
Well, it may not always. If oxygen's available, pyruvic acid is not what?
Converted into oxygen.
It's not converted into lactic acid. It's not fermented. If oxygen is not, if oxygen is not available, the pyruvic acid immediately gets fermented. If it's lactic acid, there is no transporter I'm going to caution how I'm saying this, but there's no transporter for lactic acid to get into the mitochondria. And so if we don't put pyruvic into the mitochondria, we can't do what with it? We can't intermediate step, we can't Krebs cycle, we can't electron transport chain. So oxygen won't directly be used in these initial processes, but its presence dictates whether or not pyruvic acid is transported into the mitochondria. If it's absent, Pruvic acid is going to be fermented. And once it's fermented, it's not pruvic acid anymore. Does that make sense? We've changed it to something different. So, where I'm cautioning you is that although we know glycolysis, the intermediate step in the Krebs cycle, anaerobic, they're hands down anaerobic. When you go to some other classes, they may fundamentally say, well, couldn't you argue that the linking step and the Krebs cycle are kind of aerobic? You could argue it, right? It's like, well, it doesn't, by technical definitions, it doesn't use it. But if oxygen's not available, they're not going to proceed. And so fundamentally, some people may say, they may categorize that intermediate step in the Krebs cycle as aerobic. Okay, so, and they could argue it, right? You could argue it. We could argue it all day long. Technically functional, right? So, but if we go by the literal law, or not law, but definition, intermediate step is what? Anaerobic. Krebs cycle, also anaerobic. The only true accepting of electrons happens at the very end of the electron transport chain, where we see the formation of water. So that is a survey. We haven't even talked about ATP during the electron transport chain, so we'll flesh that out in just a little bit. But I want to go back and just kind of review the highlights of each of these phases with the little summary table that you have in your guided notes. So just to recap what we mentioned, we said that we are going to oxidize or catabolize glucose. And I have in italics, or I guess it's oxidize. What is it? We said that means we're taking off its electrons. We saw NADH, FADH2 coming into play. And it's not, are we 100% efficient in everything we do? No. But it works for us. We've been living for however many, right? Our species has prevailed for a while using these pathways. So it's as efficient as works for us. At the very end, we're going to form ATP following the electron transport chain. A lot more than what we saw in the glycolytic pathway and in the Krebs cycle. The three major pathways, and you'll generally see it lumped together as three, omitting something kind of important. What is omitted here? Well, let's name them. Our three major pathways.
The intermediate link.
The intermediate, or the linking step, is often overlooked because it doesn't make for us what? It doesn't make for us ATP. Okay? It doesn't make any usable ATP, so it's often kind of overlooked. Sometimes it's even lumped into the Krebs cycle, which is, it's not the Krebs cycle. It happens, it precedes the Krebs cycle. Very good. And so I have the three major pathways. The first is what? Glycolysis. And then second would be the Krebs cycle and the electron transport chain. And I'll have a star in just a second, but between glycolysis and Krebs cycle, put a big star. Don't forget what? The linking step. So where did we say? All right, instead of, I'm going to put out some rhetorical questions. I want you to check your work, and then we'll review it, okay? So the first thing in your box, in your guided notes, glycolysis. I want you to write down where that process occurs. Rhetorical, so I want you to, everybody's going to, we'll check it together as a group in just a second. So just do your own work, check yourself. Glycolysis. What is the starting molecule? Write down what the starting molecule for glycolysis is. Write down what the final carbon-containing molecule is in glycolysis. Following glycolysis, what's the end product? So we talked about the location, the starting molecule, if you will, the final product is. Now we're adding how many net ATP? How much ATP is made? And then mark how many NADH are made. Now mark the type of phosphorylation. The final note you can add is whether it's aerobic or anaerobic. Real quick, turn your name up, check your list. You can. I didn't in my notes, I didn't speak it, so I wouldn't. It's important. Okay, let's check yourself. Where does glycolysis occur?
Cytosol.
Very good. What is the starting molecule? Glucose. The final kind of carbon-containing molecule is? The carbon-containing molecule is? Pyruvic acid, or you could say pyruvate as well. Okay, so our starting molecule, start with glucose, we're splitting glucose, and we did quantitate it into 2 pyruvic acid molecules. How much net ATP is made? 2. What do I say, net? 2 are invested, and we really make 4. Okay, so we make 4, we use 2, so that gives us a net of 2 molecules of ATP. How much NADH? 2 molecules of NADH, and also hydrogen ions. Yes, 2. What type of phosphorylation? Substrate level. Very good. And aerobic or anaerobic?
Anaerobic.
Anaerobic. Very good. If oxygen's available, what happens?
It continues
It goes
into the intermediate linking step.
It, what is it?
The pyruvic acid.
The pyruvic acid is transported into the,
Linking step.
physically it's going into the mitochondrial
Mitochondrial
matrix. Very good. Where during the linking step, it is converted into what? Acetyl-CoA. The CoA stands for coenzyme A. We didn't review its structure, but it's a coenzyme like we saw with NADH and ADH2. So once in the mitochondrial matrix, and once acetyl-CoA is produced, we can now initiate a full round of processing through the Krebs cycle. Test yourself. Do y'all want to do it per turn or totally? Per turn? Okay, so we'll go per turn. How much ATP is synthesized? Again, rhetorical, so you're going to write on your work. How much ATP is synthesized per turn? Also mark how much NADH is made per turn. Also mark how much FADH2
So what do you mean per turn? Do you mean like per glucose molecule?
per pyruvic acid molecule. If I were to talk about per glucose, I would say maybe totally. Or totally. You can also mark how much carbon dioxide is released per turn. You can mark the type of phosphorylation that would happen for that production of ATP. And finally, whether the process is aerobic or anaerobic. So how much ATP is synthesized per turn?
One.
1 molecule of ATP per turn. How much NADH? 3.
Oh, wait, you said NADH, I think. 3.
3 NADH. How much FADH2?
1.
1 FADH2. How much carbon dioxide?
2.
2 molecules of carbon dioxide. I don't have that on there, sorry. What type of phosphorylation would this be? Substrate level phosphorylation. And is the process aerobic or anaerobic?
Anaerobic.
Anaerobic. We already discussed previously that following the Krebs cycle, we've taken as much as we're designed to take of the potential energy out of the starting glucose molecule. We've got it picked up and in transit in the form of the NADH and the FADH2. So now we are going to go use the potential energy from NADH, FADH2, and then those hydrogen ions that have been liberated. So we're going to now turn our attention to the which process? The electron transport chain. In this process, it occurs across the inner mitochondrial membrane. So there's a lot, it's physically occurring across the membrane. So there's something happening in the matrix, across the membrane, and across in the intermembrane space. We are no longer taking, let me restate that, we're no longer trying to pick off energy. We're actually now going to use that energy which we've collected. So we're going to use NADH and FADH2 and their hydrogen ions. If we look at the total amount of ATP synthesized, it's going to give us way more ATP. And notice I have the about 28 plus 28 molecules of ATP. That number is variable, but without getting into the specificities, is that more than 2 or 1? They're way more. And so it's going to be, as we refer to, more efficient in the total number of ATP synthesized, but we're going to have to go through a little bit of brain work, if you will, to kind of understand how it works, right? 'Cause it's not just directly transferring a phosphate from one molecule to another. So what type of phosphorylation is it then? Oxidative phosphorylation. And the process is aerobic or anaerobic? Aerobic. We're going to see good old oxygen finally, after all this, come into play. That is a super condensed crash course of metabolism. We did the initial introduction, kind of repeating it here in the text, and now I want to go back it up with just some images to show some of the pathways. Maybe add any additional notes if we need to. And so the first process that we're going to carry out is glycolysis. This should be figure 3.15a in your textbook, simplified. Again, just reiterating that glucose is broken into 2 pyruvic acid molecules, producing for us 2 molecules of ATP, and then also 2 molecules of NADH and hydrogen ions. Okay. We also noted that the NADH and the hydrogen ions will probably be used for oxidative phosphorylation. We're going to assume they are. The ATP, what could we do with it? What can we do with that?
Use it for energy?
Use it. For some type of work that we introduced last week. So chemical, mechanical, transport, whatever cell this is in, we're making ATP, so we're making it to use it.
So it has to be like the ATP
The ATP is not transferred like you would find pyruvic acid or the NADH or the FADH2. You may recall the structure of the ATP, where those terminal phosphates are so repulsive. It's not going to stay stable very long. Right? Within like 30 seconds or so, it's going to be stable, and then it's got to be used. And so we don't want We can't store it, we can't use it. And really the whole point of these processes is to make ATP. And so the point of glycolysis, well, there's 2 points. One, quick source of ATP. So if I'm running, it's always figurative when I say I'm running. If I were running, right, my muscles would need ATP right away, because we're going to learn your skeletal muscles use probably trillions of molecules of ATP. So as much as we can get quickly, the best, right? And so even if it's just anaerobic glycolysis. So the point is, we don't want to store the ATP, we want to make it to use it. But that kind of ties us over until we can continue to process the pyruvic acids. Again, with my analogies, this is like, for your cell, the energy, like the chips at the restaurant. But you're waiting for what? The bigger payoff, right? So this will satisfy you in that moment, but there's more coming by processing pyruvic acid. Okay. So to answer your question, no, the ATP, once it's made, we use it. But the pyruvic acid can still be processed to extrapolate more potential energy. If what? If, my big if then?
Oxygen is available.
If oxygen is available, that pyruvic acid is transported into the mitochondrial matrix, which, when available, it will initiate the intermediate step. This figure 3.16a is representing the intermediate step, kind of also oversimplified. We're starting out with our pyruvate. And this here, this CoA, the C doesn't stand for carbon, it stands for coenzyme A. Okay. And this coenzyme A is a really big structure. So instead of having them put this big confusing structure so you don't overlook the pyruvic acid, they just abbreviated it CoA. Okay. Coenzyme A is bound with pyruvic acid. We get rid of a hydrogen ion to form what? Pick off a hydrogen ion and form NADH. Also release hydrogen ion. Now we see our first source of what? Let's mark this about the intermediate step. We see our first source of carbon dioxide. What happens here?
Goes into the blood.
It goes out of your mitochondria and your cytoplasm, right out into your blood, and then where?
Here.
To your lungs, and then out in the atmosphere. So it's this is where some of the carbon dioxide that you exhale, where it originates. Mitochondrial matrix following the intermediate step, or the linking step. So we started with 3 carbons, we take 1 off. The resulting molecule is going to have how many? 2. Again, this is just coenzyme A. So that forms something called acetyl-CoA. So the intermediate step makes for us acetyl-CoA. It also gives us some NADH and hydrogen ion, and also carbon dioxide. How much ATP is made here? None. That's why it's...
So does this use any energy?
By all technicalities it does, but you don't see any ATP directly coming in, but it's enzymatically regulated.
So the first 2 ATP are made, do any of those play a part into pushing it into this step?
No. Those 2 ATP, that was kind of the question asked a moment ago. Those ATP that were made don't push the pyruvic acid, if you will, into the mitochondrial matrix. There's just a transporter. Remember, we drew our transporters across our outer inner membrane. The mitochondria, it's just physically just diffuses down its gradient. Okay, so, but all technicalities, I mean, if you really wanted to go count energy transfer and everything, technically, yes, there's energy used, but not ATP, if that's what you meant by energy. No ATP is used. This is also an anaerobic process, and is priming us for the production of acetyl-CoA. But how many times did the intermediate step happen? Twice, right? If I were to clarify, first starting molecule of glucose, if I make 2 pyruvic acid, each could be manufactured into their own acetyl-CoA. So really, the intermediate step, you could double this. 2 NADH and hydrogen ion, 2 carbon dioxide, 2 acetyl-CoA molecules. acetyl-CoA, once produced, is available for driving the Krebs cycle. Figure 3.18a also oversimplifies the step. I don't want you guys to get lost in the details. When you go to biochem, etc., cell biology, you're going to procedure-wise, right, through these. But for right now, we're interested in just the concept, the ins and the outs, okay? So once acetyl-CoA is available, we can complete a turn of the Krebs cycle. Reminding you, we actually produced 2 molecules of acetyl-CoA, so the Krebs cycle could occur twice. So we could double everything up. But we studied per turn of the Krebs cycle, we're going to produce even more NADH and hydrogens. Now we see for the first time FADH2. We hadn't seen it yet. We also remove a couple more carbon dioxide, so we basically got rid of all the carbon. So as we said, we're kind of done at this point, and we are going to produce 1 molecule of substrate by substrate level phosphorylation, ATP. Same thing with the ATP. It can be used by the cell, but it can't be stored, and we can't store ATP because it's reactivity. The carbon dioxide, likewise. It's going to diffuse out of the mitochondria, into your blood, out in the atmosphere eventually. But here, now we can finally turn our attention to NADH, FADH2, and our hydrogen ions. So we've been building up to here. Are y'all ready to go there? The electron transport chain? At least one person is, that's good. So the electron transport chain is complex. I'm going to try to simplify it so that you can appreciate what's going on. There's really 1, 2, 3, 4. There's really 4 things, if I were to try to simplify it, going on here during the electron transport chain. Without getting lost in the middle, what's our end goal here? To make ATP and make a lot of it. Right? So the end goal is to make ATP in abundant amounts of it. Okay? We are going to use the NADH and the FADH2, which was just collected following glycolysis, intermediate step, and the Krebs cycle. Okay? They're ready to go. If you recall from our simple description Tuesday, those things are just alternatively oxidized, reduced. Oxidized, reduced. We have just reduced them, so now we can oxidize them, use them. Okay? How they are used are those little proteins that I had you guys draw in your inner mitochondrial membrane a week ago, and again on Tuesday. Okay? These proteins, some are embedded in the inner membrane. So if you're looking at this image, to put it in perspective, the inner mitochondrial membrane is shown here. The intermembrane space is where the greater concentration of hydrogen ions are. So not shown would be the outer mitochondrial membrane. And then on the lower portion of the image would be the matrix, the mitochondrial matrix. Okay, so you're kind of okay with me with the landmark? I'm moving back and forth here. These proteins, these membrane proteins that I had you guys draw last week, are shown in the purple blobs. Okay, so we got some integral membrane proteins. What they do is they're going to transport hydrogen ions. The hydrogen ions are shown in the blue spheres. Are the hydrogen ions equally distributed between the 2 solutions? Between the intermembrane space and the matrix? No. Where is there more? There are more hydrogen ions in the intermembrane space and fewer in the matrix.
So transferring from the membrane space into the matrix?
Well, it's actually both are going on. But first, what happens is we got to create a gradient. So we need to concentrate the hydrogen ions in the intermembrane space. So if I want to move something from where there is less of it to an area where there is more of it, if you've been in lab, what type of transport is that? Active. Okay, it's active. If I need to move something against its gradient, I need to give it energy. Does it make sense to use ATP here because we're really trying to make ATP? So we don't want to use ATP to make it's a wash.